Hello again. We understand that NCDs, such as cardiovascular diseases, cancer, diabetes, and lung disease, are the most common causes of death worldwide. The remaining 35% of life-threatening diseases are communicable or contagious, infectious diseases. These diseases are caused when a bug, a microbe, or germ invades our bodies and hijacks our systems to wage war on us. These bugs can be bacteria, parasites, fungi, and of course my favorite, viruses. All of these are pathogens, organisms that cause disease. The term is from the Greek, pathos meaning suffering, and genus, the producer of. When pathogens enter our body, they replicate, producing millions of offspring that upon contact can spread to other people, infect them, and launch a new round of disease. Clearly, the best way to deal with the threat of pathogens is to avoid getting infected. Like in many cases of attack and defense, the first thing we need to do is to be sure that we've correctly identified the enemy. Robert Koch, the father of bacteriology, formulated a system for the identification of pathogens. After graduating medical school, Koch volunteered for service as a surgeon in the Franco-Prussian War. After the war, in 1872, he was posted as the district medical officer for Wallenstein in Germany. Here, he was confronted with what appeared to be an epidemic of a contagious and devastating disease in sheep. Eventually, Koch identified the disease as anthrax, caused by the bacterium Bacillus anthracus. Koch and his colleagues knew that when blood of a sick animal was injected into a healthy one would duplicate the disease. Microscopic examination showed that the blood of infected sheep contained small, rod-like bacteria, bacilli. Could these be the cause of disease? Koch set out to isolate these suspicious microbes. At first, he proceeded to culture them in the aqueous fluid of ox eyes. Later, he found he could grow them on potato slices. Gelatin was the next substance to be tested as a solid surface upon which he could culture bacteria. But this turned out to be disappointing, as gelatin melts when cultures were attempted at body temperature. Here is where Angelina, the wife of Walter Hesse, Koch's assistant, came to the rescue. Legend has it that one summer afternoon, Walter and Angelina were having a picnic. He noticed that the jellies and puddings that she had brought along didn't melt, despite the warm and soothing sunshine. "What amazing jelly this is," Walter exclaimed. And Angelina told him about agar, a jelly-like substance made from seaweed used in Far East cooking. The rest is history. Ever since that warm summer afternoon, agar has been the material of choice to culture bacteria. The next improvement was made by Julius Petri, another assistant in the Koch lab. In 1887, Petri published his article entitled Eine kleine Modifikation des Kochschen Plattenverfahrens, a small modification of Koch's plate method. This plate modification, flat, round glass plates, are used today in all microbiology labs and have rightfully been named Petri dishes. Koch ultimately formulated a system of 4 steps that must be followed in determining whether a suspect microbe can be considered the cause of a disease. These steps are known as Koch's postulates. First, the specific pathogen must be found in abundance in every case of the disease. Next, the pathogen must be isolated and subsequently grown in pure culture, a culture that contains only one type of microorganism. The third postulate requires that the disease is reproduced when a healthy susceptible host is inoculated with the pure culture. Finally, the same pathogen must be recovered from the experimentally infected host. Koch went on to apply his approach to discover the causes of cholera and tuberculosis, and for these discoveries and research, in 1905, he received the Nobel Prize. Now, let's go to the bench and see just how bacteria are cultured on agar petri dishes.